Hello, I'm Dr. Adam Green. I'm an attending pediatric neuro-oncologist at Children's Hospital Colorado. My laboratory-based translational research focuses on pediatric high-grade gliomas, and today I'm very appreciative of the opportunity to talk to you about our work on pediatric radiation-induced gliomas, or RIGs. So as background on RIGs, we use cranial radiation to treat a variety of childhood cancers, including primary brain tumors and some higher-risk leukemias. Unfortunately, secondary tumors can be caused by cranial radiation, and the most severe of these is RIG, which is a type of pediatric high-grade glioma. RIG is incurable and even more poorly responsive to treatment than primary pediatric high-grade gliomas. Children seem to have a greater risk of developing RIG than adults after exposure to cranial radiation. National registry data suggests that about 4% of childhood brain tumor deaths are from RIG, and we think they may be even more common than this because some very late deaths after treatment for other brain tumors may be misdiagnosed as late recurrences when they're actually RIG. Considering its rate of occurrence, RIG has received very little research attention, but one prior study from my colleagues Andy Donson and Dr. Nick Foreman suggested that gene expression in RIG was very different from that seen in primary pediatric high-grade glioma. We undertook a project to understand the biology of RIG. By presenting our early findings nationally, we were able to develop collaborations with St. Jude and the Childhood Cancer Survivor Study, who were also working on this topic. We hypothesized that RIG is molecularly distinct from primary pediatric high-grade glioma, and so responds differently to treatment. We set goals of developing a comprehensive understanding of the molecular features of RIG and using RIG patient-derived cell lines from our institution, which were a world first, to understand susceptibilities to treatment. Our paper on this work, which documents the largest set of RIGs ever published by a factor of almost 3, was just published in Nature Communications. First, we found that RIG arises after treatment for a variety of childhood cancers as expected. We also found that RIG mostly occurs 5 to 12 years after the initial radiation treatment. We conducted a set of analyses on our RIG samples, including methylation array with copy number analysis, DNA sequencing, and RNA sequencing. Those results are summarized here. We found some clear patterns in our results, including a high rate of PDGFRA copy number gains and missense mutations, NF1 nonsense and frameshift mutations, and BCOR and CDKN2A copy number loss. RIG also very rarely had mutations common in primary pediatric high-grade glioma. There was only one H3F3A mutation, one IDH2 mutation, and no other classic pediatric high-grade glioma mutations. Our expression analysis showed that RIGs cluster into two groups, mostly separate from primary infant, pediatric, and adult high-grade gliomas and glioblastomas. The two groups seem to have different biological characteristics. We performed DNA sequencing both on the RIG samples and, for patients where available, matched blood samples to look at patients' normal DNA. We found that while the rates of germline mutations were similar between the two groups, the group B tumors had a much higher mutation rate in the tumors themselves, suggesting that one of the mechanisms by which group B tumors form is through impaired repair of DNA when these patients are exposed to a treatment like radiation that causes mutations. Finally, we did both predictive analysis of drug sensitivity based on gene expression and drug screening in world-first patient-derived cell culture models of RIG. We found a number of common hits between the two analyses, including DNA damaging agents, proteasome inhibitors, and MAPK pathway inhibitors, all of which are already FDA approved. To summarize, we found that pediatric radiation-induced glioma generally arises 5 to 12 years after cranial radiation for various pediatric cancers. We saw that the molecular features of RIG are mostly different from those of primary pediatric high-grade glioma, and we found that RIGs divide into two subgroups that seem to have different oncogenic mechanisms. Finally, we saw that RIGs have potential sensitivity to DNA damaging agents, proteasome inhibitors, and MAPK pathway inhibitors. We're now following up on this work to try to move toward more effective treatment and perhaps even prevention of RIG. We're trying to understand if RIG patients have genetic risk factors that we can identify before these patients get their original radiation. We've also now developed world-first patient-derived animal models of RIG that we're using to further study potential RIG treatments to move toward a clinical trial very soon. Finally, we're working to develop a consortium of RIG researchers and a RIG registry because we know the best way to understand RIG or any rare pediatric cancer is to work together. If you're interested in getting involved in this work, please email me at adam.green@childrenscolorado.org. And for the full details of this research, you can find the link to our published paper in the video description. I'd like to acknowledge our whole pediatric brain tumor research team, especially John DeSisto from my lab, who was the first author on this paper. I'd also like to thank our funding sources and our collaborators at St. Jude, especially Drs. John Lucas and Brent Orr. And thank you very much for your interest in this topic.